Now, this has become a very popular area. There have now been dozens and dozens of studies done. In fact, I keep a stack of all the correlative studies that are being done according to the Barker hypothesis, and I finally had to toss them away. I put them in a computer, but it's really quite remarkable. Literally thousands. If you cross-reference Barker hypothesis, you'll come up with 10,000 references now. Almost all of them in the last 15 years. But the studies that have been replicated and widely accepted, there's an association with small birth weight, show this. Just read them. That's an index of all the things that adult interns have to put. These are ones that are less well replicated, okay, but seem to be associated with small birth weight. Look at this: depression, behavioral problems, schizophrenia. These are associated with large birth weight. Now, it's not the birth weight, really, that makes any difference. The birth weight, frankly, is an indicator of your genetic potential, and it turns out that about 20 to 30% of your genetic potential is determined by your gene codes. Okay? Only 20 to 30%. The rest have to do with metabolism, endocrine function, fetal nutrition, and we actually know that a lot of this probably has to do with the maternal environment. That, I hesitate to show this to a group of science writers, but this was a pretty interesting cover of Time magazine in which Anne Murphy-Paul actually has really gone around the country and begun to actually delve into this area. She wrote an article, she actually wrote a book which is quite interesting called Origins, How the 9 Months Before We Were Born Shaped the Rest of Our Lives. I'm not related to her, and I have no interest in whether her book sells. And she really redacted that in the Time magazine. And so, evolution actually, you know, may be causing, in a very real sense, other problems because it was set up to just do the things that I talked about. So the real question is can we reprogram genetic programs determined prenatally or shortly thereafter? So let's say this is going on. Is there something we can do about it? Well, many years ago, I was involved in the study. I didn't quite understand the significance of it. I'm an expert in sickle cell disease, and I've been in genetics since the early 70s. And we found a drug called hydroxyurea that had a remarkable effect on patients with sickle cell disease. And just to very quickly remind you, sickle cell disease is a mutation in the hemoglobin gene. And the mutation occurs here, and in the deoxygenated state, you have a pocket on the opposite side of the tetramer. And that allows these hemoglobin molecules to line up very closely. This leads to what we call polymer formation. This leads to the red cells becoming very rigid. And this leads to occlusion of blood vessels in sickle cell disease. And the most common things people talk about in sickle cell patients is they get recurrent pain. Now, the very interesting thing is that children born with sickle cell disease really don't have any symptoms until they're actually around 6 to 12 months of age. And the reason for that is before birth, you actually make gamma, which is the fetal form of the beta globins. And there is a switch that occurs and begins to occur around 24 weeks of gestation. And then a totally different set of genes take over called the beta genes. And this switch actually is such that there is plenty of fetal hemoglobin around for the first 15 months. These two genes, by the way, are right next to each other, sort of liberally speaking, in the genome on chromosome 21. This is the gene that controls gamma. And this is the gene that controls beta. If there's a mutation in beta, such as here, okay, then one sees these types of hemoglobin molecules forming with, I just sort of put by way of a cartoon, a little yellow box there to say that's where the mutation occurs. And there is another totally different kind of hemoglobin made from 2 gamma genes and 2 alpha genes. We'll call this another form of tetramer. That's fetal hemoglobin. As your fetal hemoglobin goes away, then these can line up and polymerize, and that makes you have sickle cell disease. If for some reason you still make fetal hemoglobin, or you turn fetal hemoglobin on, then these molecules cannot line up. And therefore, actually, you can cure sickle cell disease simply by increasing fetal hemoglobin. And this is the sort of switch we talk about. Here's the fetal genes. Here is the beta S gene. Here's the normal switch that you see. And in fact, we've now found several different classes of drugs which will turn beta S down and turn fetal hemoglobin up. We are turning on one set of fetal genes and turning off a set of adult genes. And it turns out the fetal hemoglobin works just as fine in normal individuals. So this is what a sickle cell patient looks like. This is a peripheral blood smear. I keep this smear for sort of nostalgic shapes because I used it to trick my mentor, who taught me everything about red cell morphology, and he did not know that these are the same individuals. This is pre-hydroxyurea, sickle cells everywhere, and this is post-hydroxyurea, very few red cell abnormalities. This was the phase 1 study we did at Hopkins, showing that you could actually take patients and bring their fetal hemoglobins from around 4% on average up to around 16%. This was the control clinical trial that we performed back in 1995, where we randomized patients who were older than 18. We had to terminate the study early. Actually, the taxpayers gained and Hopkins lost about $1 million because they actually terminated the study because the effect was so dramatic. And there was a reduction in crisis, a reduction in hospitalizations, a reduction in transfusions, a reduction in chest syndrome, which is one of the more dangerous complications of sickle cell disease, on the patients who were on hydroxyurea. And so what we've now done is we've reversed the switch with this drug, hydroxyurea. Now, if you think about that, what we've done is we can, not can, turn off the expression of an abnormal gene and turn on the expression of a normal gene. Okay? And I would suggest to you that if people think gene therapy is substituting a normal gene for an abnormal gene, and everybody thinks it can be done physically, well, I'm showing you that we now have an FDA-approved form of gene therapy, and it's been around since 1995. We simply substitute a family member that can do the same thing for the abnormal mutated gene. So the FDA actually approved gene therapy, a fairly effective one, back in 1995. Now, what else can we do to the fetal program? Well, here's something very interesting. Many years ago, we had no idea how to feed premature babies. And the whole idea was, this is actually back when I was early in training. I'm embarrassed to say that I've trained 4 of the speakers that you've had today. Okay? And not that they're not good, it's just that it says something about how long I've been at this. But we didn't know how to feed premature babies. And the idea was if they grew faster, that was probably good. And so there were several randomized clinical trials done with prematures in which we gave half the prematures actually diets which were very high in protein and carbohydrates, and we randomized the others to either breast milk or normal milk that mimicked breast milk. And we actually showed that you could actually make babies grow faster. Now, Dr. Singhal and Dr. Lucas now started going back and looking at these trials. These children were randomized for only 4 weeks. Once they hit a certain weight, they stopped. So all that was happening is we fed babies differently for 4 weeks. Okay? Nothing more. Study stopped, out random population. But he looked at them 13 to 16 years later, and what he showed was this phenomena called split proinsulin levels were higher in the people who were given the very high protein, very high carbohydrate diet. And it was actually around 25 to 30% higher. He went on to show that one of the regulators of appetite and fat, and Fred Wansford can tell you more about this, but he went on to show that the leptin to fat mass ratios were very much higher in the kids who only for 4 weeks were given an accelerated or high protein, high caloric formula.